This is to show how important these two hormones or growth regulators are in plant development. Okay, so if you alter the ratio, as I show you, you can then get either shoots, roots, none of the above. You can also get what is called teratomas, etc. And opines are special amino acids. These are only made by, encoded by Agrobacterium tumefaciens. And these opines can only be used by agrobacteria that carry the TI plasmid. There is no other bacterium that can use these amino acids. So these are very smart bacteria. Okay, so what is auxin? So auxin is, as I mentioned, a plant growth regulator, and is made from tryptophan. We just studied the regulation of tryptophan synthesis, attenuation, pressure, attenuation, etc. So you have two sets of enzymes that are involved in converting tryptophan first into indole acetamide and then into indole acetic acid. So this simple molecule here is the potent regulator of plant growth, as you just saw. Yeah? Now, cytokinins involves AMP, and other components, and usually these involve pathways of nucleotide synthesis. And here's one typical cytokinin called zeatin. Again, it's not a complex molecule. It's a little more complex than the auxin, but it's a very important growth regulator and tends to regulate cell division, nuclear division. So, as I mentioned, not all agrobacteria in a population contain the TI plasmid, and the reason is that there is a selective disadvantage because there is another bacteria, another species in the soil that produces a toxin called agrocin that will kill. So agrocin is the name of the toxin and kills agrobacteria that contains the TI plasmid. Okay, so now we saw what we have present in the tDNA, which is the region that the bacterium is going to transfer to the plant. And I'll come back to the tDNA because none of this information in the tDNA, let me go back here, none of these genes are expressed in the bacterium. None of them. They are only expressed in the plant when the agrobacterium transfers this region to the plant and it integrates in the X chromosome in the plant. Okay? So that poses some very interesting questions about regulation. Okay, because regulation, regulatory sites and recognition of these sites varies, is different between bacteria and eukaryotes. Okay, so I'll come back later to the point. But now I want to talk about the organization of the Vir regulon. This is another very important region in the TI plasmid. Okay, so what is a regulon? A regulon is more complex than an operon because a regulon consists of genes or genes and operons or only operons. Okay, and they are all regulated by a common regulatory protein. One protein will regulate all of the operons or all the genes that make up a regulon. And this regulatory protein will recognize a consensus regulatory site in all of these genes or regulons or combination of both. Okay, so the different genes and operons that constitute a regulon may be adjacent to each other, or they may be in different parts of the genome. They do not have to be together. But in this case, they are together because they are part of a plasmid, okay? And the plasmid has, it's just not that big. So, okay, so the vir or virulence regulon consists of vir A, vir B, vir G, vir C, vir D, and vir E. And I'll tell you which ones are genes, a single gene, and which ones are operons. The squiggly lines here show the direction of transcription. This is opposite this, and then most of them are rightwards. There is VRC is leftwards. Okay. So what are all these things? What do they code for? That is what we are going to study next. So VRA and VRG code for single protein. So there is a single gene here. This is a single gene. And these are two very important proteins. VRB, VRC, VRD, and VRE are operons. Therefore, they have more than one gene, at least two genes, to make an operon. You have to have two genes at least to make an operon. And as we will see, VRB contains many genes. So VRB is an operon that codes for 14 proteins. So this is a very large operon. VRC codes for two proteins, VRC1 and VRC2. And VRD codes for four proteins, VRD1, VRD2, VRD3, and VRD4. Okay? And VRD4 is a very important protein, similar or analogous to coupling protein during conjugation. And VRE is an operon that contains three proteins, VRE1, 2, and 3. And these are very important proteins and going to play a major role in the plant cell. Okay, so VRG is here. It's a very important protein, and it is the regulatory protein. So it is regulatory protein that regulates transcription of all these genes and operons. Okay. And VRG recognizes VRB or VIR boxes, which are located next to the promoter region. VRG helps RNA polymerase bind to the promoter. So what kind of regulatory protein is VRG? Is it a repressor or is it an activator?
Activator?
Activator, good. Because it's helping RNA polymerase bind to the promoter and transcribe genes. So VRG is an activator. So the VIR regulon is under positive control, okay? Regulated by an activator, so that's positive control. So let's look at this region here, expand this region to look more closely, and we'll see that in front of VIR A, we have a VIR box, we have the promoter, the VIR A gene. In front of the VIR B operon, we have VIR box, promoter, and the operon. Okay? There may be, in different cases, more than one Vir box. Okay? But to simplify things, this is a cartoon that expresses that you have a Vir box, a promoter, and a gene or an operon. All of them are going to be then regulated similarly. Okay? And to expand on that concept, here we have Vir A gene, the promoter with RNA polymerase bound here, and then Vir G interacting bound to the Vir box and interacting with RNA polymerase to regulate transcription of this gene. So transcription starts and stops there. Here you have again Vir G binding to the Vir box, interacting with RNA polymerase and transcribing the many genes present in this operon, and so on and so forth. So this is how Vir G is regulating expression of these genes and operons. So as I mentioned, some operons in the Vir regulon contain more than one Vir box. Are there any questions so far? Okay.
Dr. Benuet, I'm sorry, I'm trying to unmute myself. This is Mike. When you say that there's more than one Vir box, do you mean more than one before the promoter on each?
Yes. So there could be like 3 or 4 of these things right next to the promoter.
Oh, okay, thank you.
And that has to do with also, then you will have multiple VRG molecules binding, and will have to do with the strength of the promoter, how much that region is transcribed. So it gets into subtle questions of how each of these is regulated, because not all of them are regulated to the same extent. In other words, you don't make as many transcripts from each of these regions, but these are subtleties of regulation or transcription.
Okay, and with the ones that have more than one Vir box, are those harder to activate than the other ones that have only one Vir box?
No, actually not, because then this is going to be the VRG protein is not going to be a limiting factor. It's going to be abundant. So as long as there are boxes to bind, VRG will bind, and RNA polymerase is a very abundant protein. So they are not limiting factors.
Okay.
Okay, so then we have, I will come back to the Vir regulon, the VRB, VRB operon. I'll come back to tell you what does it code for. But first, I then want to take the question, how does Agrobacterium tumefaciens transfer the tDNA to plant cells? So Agrobacterium infects only wounded plants. Okay, this is really key. Okay, and I mentioned this earlier. And so wounded plants, what happens when a plant is wounded? And plants are wounded either, in farming by machinery, or a person walking by a plant, or by insects or other kinds of insult to the plant. When a plant is wounded, they produce compounds, phenolics, so these are derivatives of phenol, that protect the plant. They fight invasion by pathogens. They also produce certain sugars too that are part of this wounded response of plants. And this is very important in why Agrobacterium infects only wounded plants. So wounded plants produce phenolic compounds as a defense against microbes. And this is a response basically of all plants. When you wound them, they have this response. And phenolics normally are red color, purple or dark purple color compounds. Okay, and here you have some of these phenolics produced. Okay, so they are all have the phenol ring and then additional moieties attached to that, and these are all phenolics that are produced by plants. This is inactive, the active form, etc. And some of the sugars that are produced in this response too. So one of the phenolics that is produced is acetosyringone. And let me tell you that this part of this work, was elucidated by an undergraduate doing research on this organism. So acetosyringone looks like this, simple, just phenol with these groups here, there's metal groups, okay? And the plant senses, Agrobacterium senses this acetosyringone. So it senses the presence of acetosyringone, and the way it senses this involves what is called a two-component signal transduction pathway. And I'll describe that in a moment, and I'll describe the proteins involved in this perception. So let me tell you in general what a two-component signal transduction pathway is, because these play very important roles in plants, fungi, and bacteria. Animals do not have two-component signal transduction system. So a two-component signal transduction system is a pathway that transfers a signal from the outside to the interior of the cell, and this results in an output response. An output meaning you are going to have to transcribe genes that give a response. So it involves perception of the signal by a sensor histidine kinase protein. And I tell you in a moment what is the sensor histidine kinase in Agrobacterium. And then the transduction of the signal from this sensor to a response regulator, which is usually a transcription factor. And then this transcription factor is going to activate genes, and you have a response.